Oh, but you're recording before presenting, Brittany?
Okay, so hi everyone. So I'm Brittany, and this presentation is based on suppression of antiviral RNA interferase by viral 2B and B2 proteins in plant, insect, and mammalian cells. So for RNA interferase, or RNAi, it's a genetic regulatory system that functions to silence the activity of specific genes. There are three main steps to focus on within RNA interferase, which is dicing, splicing, and silencing. As you can see from the top image, we can see how the double-stranded RNA is diced into siRNA, or also known as small interfering RNA by the dicer. Then the siRNA duplexes or complexes are a guide to help slice the messenger RNA complex using RISC, or also known as RNA-induced silencing complex. So once the messenger RNA or mRNA complex is produced through RISC, the result would lead to gene sequencing or degraded target messenger RNA. So for antiviral RNA, interference is basically the host defense against viral infection, which will prevent viral infections of plants and animals. So the antiviral RNA interface approach would be used against the virus or the virus receptor, which the gene's silencing would prevent the entry to the cell. So one key point to note is that the plant and animal viruses produce viral suppressors of RNA interface to promote infection. So the RNA interface would lead to silencing genes within dicer and RISC, or RNA-induced silencing complex within the cell, compared to with antiviral RNA interface, which we would need to use dicer, Argo proteins, and R2D2 proteins in order to defend against viral infections, which are viruses trying to come in the cell. So one example of a plant virus would be cucumber mosaic virus. The cucumber mosaic virus has positive strand RNA viral genome like COVID-19, and the viral genome encodes 2B protein to block RNA interface. And then with the flockhouse virus, it's present in insects and can kill fruit flies. So the flockhouse virus has a positive strand RNA genome and encodes B2 protein to infect RNA interface. So the next virus is Notomira virus, and that's very relatable to flockhouse virus due to both viruses encoding B2 proteins. And B2 proteins are present in both insects and mammalian cells. So the mechanism of RNAi suppression by 2B and B2 proteins. So 2B and B2 proteins share no sequence similarity, so they are not homologous proteins. However, both proteins do bind to viral long double-stranded RNA. So the mechanism of these two proteins would isolate long double-stranded RNA and prevent dicer processing or dicer recognition. So the proteins would inhibit production of viral siRNA or small interfering RNA and make the antiviral RNA interference become weak. 2B and B2 proteins can replicate and infect RNA interferase, specifically when the small RNA goes to the complementary RNA for direct splicing. With this in mind, there would be no inhibition of RNA interferase, and RNA interferase would not be as efficient. So the summary of all this is that antiviral RNA interferase is the host defense against viral infection, and the host is trying to suppress the disease. However, there is a counter defense, which is 2B and B2 proteins. And if we take away the counter defense, which is the 2B or B2 proteins, which are also viral suppressors, then the host can win. So the gap or hypothesis would be to use RNA interference mechanism to treat or prevent blue tongue virus infection. So the methodology would be using a knockout gene to knock out B2 proteins, since the blue tongue virus is for mammalian cells, and by using the knockout gene, you can activate the cell to use RNAi. Another thing would be to use live cell imaging, since this gene, the knockout gene and the virus is going to still be in a mammalian cell. We can use it to track gene silencing in real time and reveal the variability of RNA processing. So the procedure would be preparing 3 groups of healthy mice, and then 1 group of infected mice with the blue tongue virus would need to have the B protein would need to have the knockout gene of B2 protein in the viral genome. And then another group of mice would just be infected with blue tongue virus. Another group of non-infected mice would be injected with corn oil, and we would use RNA interference to make primers with dicers to specify which strands are going to be cleaved within blue tongue virus. And we would monitor RNA interference for each mice group with live cell imaging, and we record the data for a month, since the symptoms of blue tongue virus are about 2 to 4 weeks. So the expected results would be preventing expression of blue tongue virus in infected hosts, and the control groups of the blue tongue virus, as well as the healthy mice who were injected in with corn oil, would have little to no reaction, or very similar to healthy or infected mice. So the implications for future research is to use the mechanism to develop different primers and or knockout genes for multiple viruses, and to potentially prevent lethality rates of viruses. We can also use the RNAi mechanism within plant viruses as well as insect viruses. And with the mechanism, we would try to create a cure or vaccine to help incurable diseases such as HIVs or AIDS. And then I would like to thank Professor Ding in the Microbiology and Plant Pathology Department at UCR, and everyone for listening to my presentation. And then these are the references.
Thank you so much for your presentation. So again, we will open either the mic or the chat for any questions.